Welcome to our deep analysis of Biology Paper 3, Kcse 2023. And this Paper 3 contains 3 questions. Let's start with the question 1. You are provided with the following materials and apparatus: a prepared slide labeled E, containing transverse section through a plant organ, access to a light microscope with at least the low and medium power objective lens. The first question, part A: observe the section under the low power and medium power objective lens of the light microscope. Roman 1, fill the portion label F in the plan diagram below to show a portion of the structures seen under the medium power objective of the light microscope, and label the structures. The portion was here as seen under the field of view, then it cuts across here. Then what was observed? It was the xylem and phloem. The xylem and phloem. So the student needed to draw this xylem and phloem. Xylem and phloem, because the structure observed, it was a stem of a monocot plant. And when filling that portion, you must use a pencil and observe certain rules, and those are the rules of drawing. Number 1, outline for epidermis, and it should be a double outline. This one, or a single outline in this case. Then a drawing mark is denied if you've drawn the xylem and phloem out of this portion. Then a continuous line for vascular bundles. Continuous line, which means it should be continuing, should not be maybe not reaching each other in layman's language. Then no shading, because in biology, the biological diagrams when drawing, we don't shade. Then orientation of the vascular bundles, that is xylem and phloem, was ignored. Then labeling, the label should be correct. The xylem and the phloem plus the epidermis, and these label lines should not cross each other, or you should not use a bullet when labeling. Also, a arrow head should not be used. Then this label line should touch the structure. For example, xylem here should touch the structure to show it is xylem. The same to phloem, should touch the structure to show that it is phloem. The same to epidermis, in order for you to get the 3 marks. The drawing mark was 1 and labeling was a 2 marks. Roman 2, calculate the magnification of the image under low power objective lens. And the magnification of a microscope is a objective lens magnification times eyepiece lens magnification, and in this case they have specified low power objective lens, and in this case it was between 5 and 15. Then objective lens magnification was between 2 to 10. The correct substitution it gave the correct answer depending with the power or the magnification of your low power objective lens and the eyepiece magnification. That means it ranged from 10 to 150, and remember, when writing the total magnification, you'll have this X here to show it is magnification. Part B, with reference to one observable feature, say the class of plants from which the organ was obtained, and that was monocotyledonae. That is class monocotyledonae. You should start with a capital M, and reason which you have said observable, which you are able to observe. The vascular bundles were scattered. In this case, you can see the vascular bundles were scattered or randomly arranged or irregularly arranged. And for you to get the reason, you must be able to give the correct class. If you give another class, for example, the Cotyledonae, it is totally wrong. Name the plant part from which the section was obtained. That was a monocotyledonous stem, and simply in a monocotyledonous stem, the vascular bundles are scattered. Part D, state 2 precautions one should ensure or should take to ensure safety of the slide during observation under the microscope. Number 1, you hold the slide on stage with clips firmly, and do not break the slide, or take care not to break the slide. Never use coarse adjustment knob with high objective lens. You use fine adjustment knob with high power objective lens. That is to prevent breaking of the glass slide. Then lower objective lens or body tubes. You lower the objective lens stroke body tube slowly to avoid breaking of slide. Use coarse adjustment knob with the low medium power objective lens. One of those or two of those are some of the precautions to be taken. Part E, state how each of the following parts of a light microscope contributed to clarity of the image of the section observed. Mirror, and the mirror reflects light onto the stage or slide or specimen or into the microscope. And in this case, you must insist on reflecting light onto the stage. Reflection of light only, it is an omission, because where does it reflect? It means it reflects only on the stage of the slide. Diaphragm, it regulates or controls the amount of light landing or falling on the object on the stage. Then condenser concentrates light onto the object or specimen or slide on the stage, or in other terms you can say condenses the light onto the object on stage. And the student must show the path of light and destination of light. For example, if it is from the mirror reflects it through the diaphragm. The diaphragm, as we have said, regulates the amount of light falling onto the object on the stage. Then from the diaphragm to the condenser, which concentrates light onto the object on the stage. Question 2. You are provided with the following materials and reagents: half a portion of raw banana, 3 beakers labeled G, H and J, treated as follows. Beaker G contains 50 ml of dilute hydrochloric acid. Beaker H contains 50 ml of distilled water. Beaker J is empty. Scalpel, a spatula or a pair of forceps, a white plain paper or white tile, stopwatch, stroke means of timing. Next, when some plant tissues are exposed, enzymes on the exposed surfaces react with oxygen. Using the provided materials, investigate the enzyme oxygen reaction using the procedure below. Now the key terms: enzymes on the exposed surfaces react with oxygen, and you are told to investigate the enzyme oxygen reaction using procedure below. Let's look at the procedure. 1. Slice off about 2 millimeter from the exposed end of the raw banana and discard the slice. The diagram is here. This part 2 millimeter from the exposed end, then you discard this part, this 2 millimeter, discard this part. Then Roman 2, slice another piece about 1 centimeter wide from the remaining banana to use in the investigation. In this case, this is the 1 centimeter wide here, which will be used. Then Roman 3, divide the portion obtained in 2 above into 3 parts, A, B, and C, as illustrated in the diagram below. You can look. These are the sliced part in Roman 2. You divide into 3 portions, that is A, B, and C. The A, B, and C portions are here for use. Let's look at Roman 4. Remove the peel from portion A. Cut the peel into 3 pieces and immediately drop all the 3 pieces into beaker G, the one containing hydrochloric acid. Obtain peels only without remnants of banana flesh. That is a portion A. You are being told you cut the peel into 3 pieces. The peel for use is here. The peel for use is here. Then you drop them into beaker G, which contains hydrochloric acid. And NB, obtain peels only without remnants of banana flesh. In this case, it is just peels only. And in paper 3, for you to get the maximum marks and correct observation, you should follow the instruction carefully. Roman 5, repeat procedure 4 with peels from portion B into beaker H, that is containing distilled water, and those from portion C into beaker J. J, we were told it is an empty beaker. So we are using C, the peels from C, we put them into beaker J, which is empty. Then peel from B, we put into beaker H, which contains distilled water. Then peel from portion A into a beaker containing dilute hydrochloric acid. Leave the setup for 5 minutes and observe the inner surfaces of the banana peels in each beaker. Record the observation made in each case. G was the beaker. We are saying G is a beaker containing hydrochloric acid, and we use the peels from portion A. The observation, we had a white or a cream or yellow color, or yellowish white, or a cream yellow, or in other terms, we accept no color change, or the color remained the same. Then H was a beaker containing distilled water and peels from portion B. The color was a brown, and also you can say gray color. There was a gray color or a black color. Then J was an empty beaker which contained peels from portion C. The color was a more brown or black, or there was a browning or blackening or gray of the color. Roman 2, account for the observation made in beakers H and J This question is tied with this one here. Kcse, water covered the surface of the banana peel, or it diluted or dissolved the enzyme, hence less or little oxygen in water, hence the enzyme oxygen reaction, leading to oxidation, resulting to less browning or brown color. Remember, the H had distilled water. So we are saying this water diluted that banana peel and therefore dissolving the enzyme, hence less enzyme oxygen reaction, resulting into less browning. Then J was a beaker which was empty, and the peels from portion C, the peeled surface was fully exposed to oxygen or atmospheric air, hence maximum enzyme oxygen reaction, resulting to significant color change, that is a more black or blackening or more brown. Those are the accounting part between the beaker J, which was empty. It means those pills containing that enzyme were exposed, fully exposed to oxygen, hence more reaction, that's leading to more blackening or more browning. Unlike in H, where we had distilled water which diluted the enzyme. Roman 6, using a pair of forceps provided, remove the peels from each beaker and expose the sets of peels separately on a plain paper or a white tile provided. Leave them for another 5 minutes and observe. Part B, Roman 1, record observation made on peels from beaker H and J. Remember, beaker H had distilled water, then J was the one which was empty. The beaker was empty. In H, it turns brown or brown color increases or accept black color and gray color. And then J, it remained brown or black. Roman 2, account for the observation on the surface of peels from beakers G and J after a further 5 minutes. G, the one which contained distilled water, oh sorry, not distilled water, but hydrochloric acid, no color change or white or yellow color, because the acid, which has a low pH, denatured or destroyed the enzymes. Then J, enzymes on the surface of the peel were fully exposed to atmospheric oxygen, hence surface was fully oxidized, turning brown or gray or browning or blackening. Or in other terms, you can say the enzymes were exposed to optimum or best pH. Then part C, suggest the suitable pH for enzymes found on the surface of banana peels. That is a neutral, or we can accept a slightly alkaline, or any pH between 7 to 9. Part D, suppose the peels in setup J were initially boiled for 5 minutes. Suggest the observation that will have been made. No color change or yellow color or cream color. Explain observation. The observation made. Reason: boiling denatures enzymes. Boiling denatures enzymes. We can move to the next question. Question 3. You are provided with specimens labeled K and L together with photographs of organisms. Label N, P, Q, R, and K was grass, L maize grain. Assuming the organisms are found in the same ecosystem. Part A, Roman 1. Construct a complete food web that includes the specimens and photographs in the organisms in this ecosystem. We have K, which is grass, and maize, maize grain. N, which is a mouse. Then Q, a bird. R, a cat. Then P, a caterpillar. And a food web just contains numerous, or it is just an interconnection of food chains, and the food chains, or in that case food web, must start with a producer. In this case the producer is a grass, and grass we said is K, specimen K. Apart from grass, the maize seeds or maize grain, that is L, are also producers. In this case we have two producers. In this case the worm or caterpillar, that is P, feeds on grass. Then the larva or the caterpillar or the worm is fed on by the hen or pigeon or dove or specimen Q here. Then the cat here feeds on the pigeon or the Q, specimen Q. Then we have the mouse or specimen or organism N is fed on by the cat, and the mouse feeds on this caterpillar. And when constructing the food web, when this crosses interchain, you do a loop. The correct way is you loop here to show that larva is eaten by the mouse. In this case grass eaten by the pigeon. Then also maize seeds are eaten by the mouse. Maize seeds are also eaten by the caterpillar, and maize seeds are also eaten by the pigeon. In this case you can use the letters or the real names. From the food web, identify the longest food chain, and that is grass, which is K, fed on by caterpillar, which is P, bird fed on by cat, which is R. Or in other terms, grass, caterpillar, rat, cat. Maize eaten by caterpillar, caterpillar eaten by bird or hen eaten by the cat. Then maize grain eaten by caterpillar, which is P, eaten by the rat, which is N, eaten by the cat, which is R. Then conditions to score on the construction food web: there must be at least a producer, which we said it is K or L, that is maize or grass. Then at least two food chains in order to score. Then we accept if the candidate draws the organism. Then small letters or capital, it is accepted. The use of dotted lines, it is not accepted. Or lines with bullet ends, not accepted. Lines without arrowheads, it is not accepted. Next question, B, Roman 1. Identify the organisms with the highest biomass, and that is a grass or specimen K, or maize grain or specimen L. Reason, give a reason for your answer, and that is because the grass or the maize grain is a producer, or in other terms manufactures own food, and this it enables it to sustain the whole ecosystem. Part C, other than feeding, explain how the organism represented in the photograph R benefits from specimen K. R benefits from specimen K. R is a cat. How does it benefit from grass? In short, importance of grass to a cat. Specimen K, which is a grass, releases oxygen used, which is used by specimen R, the cat, during respiration. Then specimen K purifies or cleans the air by using up carbon dioxide exhaled by specimen R. Then specimen K can be a habitat for specimen R. Then specimen K is a habitat for other animals that R feeds on. Then specimen K can be a hideout for specimen R to get its prey or escape from danger or predators, or it is important for breeding, or used by R, the cat, for stalking the prey. That is the importance of grass to a cat. That was a Kcse paper 3 2022. I hope you subscribe, you watch, and you like. Thank you.